Okay, this is the last lecture before good old exam 2, which you can find at Quizlet under PCB 4522 exam 2, except that's a compilation of all the homework. But it does make a good place to go to get some flashcards and be able to have a fast response. So it's all good. So let's finish up the retro virus / retro element chapter here, and then we'll take some questions and we can talk about exam 2 if you want, and we can continue this discussion on Saturday in the microbiology main seminar room at 1 o'clock. I'll be holding a review there. So let's talk about the different categories, just as sort of a review, of these retrotransposon-like elements. So you have retroviruses, right? We all know what a retrovirus is. It produces an infectious particle. You can catch one of these things. They're transmissible, and they have long terminal repeats. They encode one major transcript, but it gets alternatively spliced, and then you have frame shift or suppression and protease degradation of a pre-protein. So you end up with about 8 proteins in the infection cycle. So retrovirus. Then you have things that share similarities. The next, all these other things. So they're retroviruses and then everything else. The everything else have a name. They're called retroelements. That's kind of a broad category to cover everything except the retroviruses. The retroelements have 3 different flavors. One of those looks very similar to a retrovirus, except the particle that it makes is not infectious. It doesn't burst the cell, lyse the cell, and infect other cells. It encapsulates itself and then reinfects its own nucleus. And they are called LTR because they look just like a retrovirus. They have long terminal repeats. They're called LTR retrotransposons. Okay, and you have elements that are in yeast and Drosophila and corn and stuff that we know about. And then the last component are things that do not have a long terminal repeat, yet some of them make things that look like reverse transcriptase and integrase and things like that, but they just use a completely different mechanism of integration. They don't have any of this long terminal repeat. They're called non-LTR, but if you look carefully, you'll see they're not called non-LTR retrotransposons. They're simply called non-LTR retroposon. They left out the trans. Retroposons. Sometimes it's shortened to retroposons, meaning they do not have an LTR. And we'll see they have a very unique way to insert. And then the last category is it's not a derivative of the retroposons, but it relies on retroposons to jump around. So you can say, it can't get any more complicated, right? I mean, let's make it even more complicated. I'm sorry, but that's as much as I can do. That's as complicated as it gets. And so keep these big categories in mind as a way to sort of keep it all straight in your head. So here you see, this is the last part of a retrovirus. Notice it has let me get into the right PowerPoint mode here. The retrovirus mode, this is overlapping what we covered last time. Remember that here you see host, then you see the retrovirus, and notice the retrovirus is completely intact here except for 2 nucleotides on each side, right? This form of the virus, retrovirus, when it's integrated, is called what? A provirus. The provirus form of the life cycle. When it's integrated and it's almost complete except the 2 bases, the C and the A. CA has been cut off from either end. Which enzyme did that? The integrase itself in the preparing the linear double-stranded retrotransposon for insertion. And notice these repeat elements here, U3, R, U5, U3, R, U5. Everything is complete, right? Right here on either side, you have 4 to 6 base pair direct repeat, and this is like all transposons because the site that they insert is cut. The target is cut, and it's a staggered cut. And repair of that staggered cut, because you do it for each strand, ends up repeating the portion of the cut that is staggered, that's offset. As you repair that offset on each side, you end up repeating something that was not repeated before. So this is a familiar transposon action. And what else? Now you have it, we've talked about this complicated replication cycle. And how is you have plus RNA, it's packaged, it infects, it opens up in the cytosol, it's RNA, it's just been born. And how does it make the first DNA copy of itself, which is called minus DNA, since it is the plus strand? How does it prime that event? And this is common with all the retroviruses. It primes using something from the host, right? A host tRNA. The 3' end of the tRNA binds right there. And then goes, it always is here, and extends to the end. Well, when this is out of when this is just RNA, here you see it as the provirus. But anyway, I can't illustrate it here in the provirus form, but it goes to the end, forms this strong stop DNA, which then jumps back and hybridizes again, anneals with itself in the R region, and then is used to prime the rest full length. And you're familiar with that, I'm sure. So here you see so that's replication. Now we are talking about expression of the genes. How is the virus itself, how does it express its genes that encode the proteins that enable it to have all these enzymatic activities? It has, in the repeat, in U3, there is a eukaryotic promoter that works with RNA polymerase II of the host. That's the one that encodes, that transcribes genes that encode proteins. And so the transcript produced by RNA polymerase II in eukaryotes, it attracts a series of protein complexes that are designated to process the new RNA. What do you need to do to prepare the initial transcript that's eventually going to encode a protein, which is translated in the cytoplasm? What do you need to do to that piece of RNA to prepare it so that it's in a state that can be used on a ribosome? You have to do 3 things, actually. Usually they're full of introns, right? So you have to remove introns. So the RNA polymerase itself has a great long tail that it drags along behind it. It really is very long. And it's repeat over and over and over of 7 amino acids. And attracted to the tail, depending on its phosphorylation state, is a complex that splices, called the spliceosome. That's why transcripts from Paul II know they're supposed to be processed, because the polymerase itself has this long tail that just pulls along that all these complexes bind to. The second complex that is looking for a binding site on this tail, you know, like the baby gators, you seen those guys? They're all hanging on to the carboxy-terminal domain of RNA polymerase II. Each is a complex that's up to something. Another complex that is binding to that tail that's drug along behind Paul II is one that puts a poly-A tail on the transcript. That's the second thing you need to do to the transcript to make it work on a ribosome. And the last thing is actually very important. On the 5' end, you have to add a 5-methyl G. Is it 7-methyl G? It's 7-methyl G. It's one of the two. It's an odd number, 5 or 7. You have to cap it at a methyl G on the 5' end, and that says I'm a messenger RNA, because the eukaryotic ribosome is not looking for a Schindel-Garner. It's not looking for a ribosome binding site. It only, in its brain module, it's only looking for one thing. That's the cap. When it finds the cap, it goes, this must be a message. This is not a tRNA. It's not a structural ribosomal RNA. It's not a degradation fragment of a long piece of RNA. This is really the 5' end of a message. Hallelujah. And then it starts scanning until it finds the first ATG, or AUG since it's RNA, and it starts translating. It's always looking for the end that's been modified, been capped. Okay, so this transcript, because that's a little aside in exam module 4, but this transcript has all those properties. It's made, synthesized by the polymerase, which is designed to process a messenger RNA. So this will have a poly-A tail on the end of it. And it'll have a 5' cap because of the polymerase that has expressed it. And that's because the promoter here is one that attracts Paul II specifically. Whew. That was long-winded. This is just an introductory slide. So here you go. You have the message, and of course you get it translated like this, and there's a stop codon. You get GAG being produced. Ribosome falls off and stops. In a certain percentage, they say it's 5% in the book, you'll get GAG going all the way into Paul and envelope because of either suppression of the stop codon. How do you suppress a stop codon? I mean, stop is stop. What doesn't the cell understand about no? Stop. No, don't oh, that, too late. How does that work? There's a glycine tRNA that's there in a small abundance, low abundance, that its anticodon on the tRNA fits a stop codon. There's always that tendency to be able to read right through a stop codon. It's that built-in ability on rare occasions to do that, and the virus exploits that. That's suppression, because it uses that glycine stop codon in parentheses. Another way it reads from GAG into Paul is frame shifting. There's a certain sequence of amino acids, but underlying that, a certain sequence of base pairs, which is not very complementary to translation. It causes the ribosome to sort of stutter and get out of frame and not stop and shift its reading frame. And so that's a way you can fuse a gag onto a pol at the ribosome level. Okay, both of those occur at the ribosome level. So you get, well, this is, I'm talking about something else, and the slide's got its own little narrative going here. So here you have the RNA, and I took a little aside to tell you how different ways it's expressed. And then this, by reverse transcriptase, is turned into linear DNA. Of course, the linear DNA form, U3R, U5, U3R, U5, is the only completely 100% intact form of the retrovirus. This is before the CA and the CA on either end are cut out by the integrase before it integrates. Back to the original story here, sometimes the transcript, because it's Pol 2, polymerase 2, it's the signals that tell it to stop transcription are not well understood. It often will continue for several thousand base pairs beyond where it normally should stop. What happens is there's a signal in the RNA after translation, but the polymerase knows nothing about translation, but there's a signal in the transcript which says, I don't care whether you're finished transcribing or not, but right here is where you should add a poly A tail. In order for a poly A tail to be added, you have to cut the transcript. The polymerase now can keep on going, you could care less, but you have to cut that message right here and then get another polymerase to just add A's. 200, 300, 400 A's are added on the end. So it's not encoded in the DNA, but it's added. And the polymerase just keeps sailing along until it finally gets tired and falls off. We think. It certainly goes on. Okay. So that's kind of a summary of the retrovirus story. Let's now talk about what the new stuff is for today, and that is the ability of retroviruses to cause cancer. They pick up cellular genes and form a defective retrovirus. Because the cellular gene, and it happens to be one that if you express it in an uncontrolled manner, results in a tumor. So it's called an oncogene just because a tumor results. It's actually performing some special purpose. It wasn't put there to make tumors. It's just when it's unregulated, that's the result you get. It's called an oncogene. Could be one of several hundred different ones could be an oncogene under the right circumstances. So it picks up somehow a copy of a gene when unregulated results in tumor cell growth or uncontrolled cellular proliferation. So this gene is carried, and we don't know exactly how this works. They think it happens through a deletion event, and here you see the viral oncogene, because the retrovirus jumps next to a host gene, and then there's some recombination, deletion, where this gets fused onto the last half of the retrovirus, and then that gets turned into a transcript, and eventually that gets repaired, and you get the N back on. You can read the text, and you see it's a little complicated. But what we know is that this gene now is a part of a retrovirus, and it has the long terminal repeats at each end. It can jump, it can invade, it can insert itself into your genome, except it had to substitute all that good stuff for this viral oncogene. So it really can't do it by itself. It needs a helper. It's sort of like mini TI, right? It's like helper plasmids, that whole concept of this is carrying something. It's got the right border, left border. but it doesn't have the TRA genes or the virulence genes. They're here. It always has to have a helper retrovirus to actually cut it out and insert it someplace. A lot of times, of course, the only way this is, you're going to see this retrovirus containing this oncogene is it has to be in a cell that has the helper virus also, otherwise it can't move and it's unable to propagate. So these are always linked together, the helper virus version of the defective virus, which has picked up a cellular gene which happens to cause cancer. It's also interesting that in the mature particle of a retrovirus, you always get two RNAs packaged. One could be a helper, just by probability, just by pure chance, and the other could be the wild type, the defective, the helper, which is the wild type, and the defective one that contains the viral oncogene. If you look at the origin, we've sequenced a lot of these viral oncogenes, and if you look at the gene that was originally in the host and say, aha, this originally was that gene over here on chromosome 22. And if you look at it in its original configuration, it has introns. But if you look at it here, it has no introns. What does that mean? That means this is derived by some contorted pathway from a transcript that was processed and then somehow got incorporated here in the retrovirus. So you can think about ways that might happen with reverse transcriptase being able to start on one retrovirus RNA and then jump to another, and then by recombination based on that phenomenon end up with something like that. But it's not a simple one-step transformation into a cellular transcript, a messenger that is incorporated eventually into a retrovirus, substituting for Paul and envelope. Okay. They're initially expressed as GAG fusions because for some reason this part, the GAG part and the frame shift and all that, those mechanisms are still intact. You never really get rid of everything in here. You need some of this GAG there. And these, you remember, are matrix and structural proteins. Maybe it just works better if you have something that makes structural proteins, because if you have an enzyme, it can work over and over and over and over again, but a structural protein, one copy, it does the function. You don't get an amplification of function because it's an enzyme that's used over and over again, for example, to cut, to transpose, to integrate. Transducing viruses, that's what they're called. Transducing viruses are the ones that have the viral oncogene. And here's a scheme on how they could have arose at the provirus, and you have a deletion, and you have an oncogene right next to the inserted provirus, and you end up with this, and then you get splicing. You notice this has no right LTR, and by recombination, by reverse transcriptase, you get it packaged, and then eventually you get it back. So it has LTRs and requires a helper. I'm not going to ask you to recapitulate that on an exam. That's what the question was in all your minds. The answer is, are you kidding me? That's way too much detail. You don't really need to know that. You just need to be aware generally of how these things work. Okay, so that is the end of retroviruses. Everything that comes after retroviruses goes under the category of retroelement. So here we go with the first retro element. It also contains a long terminal repeat. It's just not an infectious particle. It makes a particle but not infectious. So this is one that you'll need to know for the exam. You need to know that Ty specifically is a yeast LTR retrotransposon. Sometimes you can leave off the LTR part, because if you just say retrotransposon, it implies it's LTR retrotransposon. Just like non-LTR retroposon, all you really need to say is retroposon. You can leave off the LTR part. But Ty, wonder what that stands for. It's a transposon in yeast. I don't know, I'll look it up. Ty, and there's a whole series 1 through 5. Yeast transposon, transposon yeast. And you don't need to know the details of this, but you can just see it has 2 open reading frames, and it needs at a minimum some enzymatic activity. That's the Paul relatedness, and the A is the structural component that's going to make this capsid that never infects but remains packaged inside the cell. So you see it's very similar. You get the 2 size messages. Alternative splicing is happening. Get frame shifting. Everything is very similar to a retrovirus, except it's not. No envelope. So what's left to package? You remember in the viral structure you have the nuclear capsid, and then you have the matrix, the nuclear matrix. You have a couple of packaging layers that are inside the envelope and the spikes and the balls up top. So this doesn't have the envelope, which is the spike and the little cap. It has the LTRs, and this is what it looks like inside of a yeast cell. If you look at on the electron microscope, you see a perfectly formed retrovirus particle, except it doesn't have spikes because it's still inside. It hasn't budded out yet because it can't. Capsid protein packages RNA, transports it to the nucleus. Not infectious. There's one in Drosophila called copia. There's some in plants too, but copia and Ty are ones that you might need to remember. These are retrotransposons, LTR retrotransposons. And I kind of remember seeing that figure on more than one occasion over the years. I can't say any more than that. Retroelements. I'm kind of backing up here, the 3 classes. Elements, this is everything except retroviruses. The LTR retrotransposons encode transcriptation integrase, not an infectious particle. Ty and copia, this is yeast, copia is Drosophila, or examples. The second are the retrotransposons, non-LTR retrotransposons, or sometimes just called retrotransposons. And they encode reverse transcriptase, nucleic acid binding protein, endonuclease. We're beginning to deviate here. What's with this? No long terminal repeats, as the name suggests, derived from RNA Pol II transcripts. That ain't right. Example, lines. Long interspersed nuclear elements. In the human genome, there is one line, which is a retrotransposon you inherit from your parents. Both your maternal and paternal have lots of these things, and you got them too. They've been with us for 100 million years. They will last, well, retroviruses are no longer active, the ones that we inherit. We have retroviruses embedded in our genomes, full-fledged infectious retroviruses, but they are defective now, and they haven't been active for tens of millions of years. I think I read that it's 50 million years ago is when they were last active. So what were we doing 50 million years ago? What campfire were we sitting around? Actually, we weren't doing a lot of sitting. We were sitting on a branch. That is when we decided we'd had enough of dating old world monkeys and decided to go off on our own and develop our own line of primates. That's when the human line, the apes separated from the old world monkeys. They lost their tail 50 million years ago. That's the last time that the retroviruses were still active. Depends on what mammal you are, whether they are active or not. Some still, like mice, still have a lot of active stuff going on. Lines, okay. So these things are mostly inactive, but we have one version that is still active. L1 is a line. And we have 80 to 100 copies in our genomes, all of us. And occasionally, they will decide to jump from one place to another. And you just hope they don't land into something critical or activate something that's going to cause a tumor. They jump about 0.3% of the time. Lines, long interspersed nuclear elements. And then, of course, signs. And this will make more sense later. Short interspersed nuclear elements. These, again, have no LTRs. They are derived from Paul 3 transcripts. So we got Paul 2 and 3. The only one missing is Paul 1. Everybody remember what Paul RNA polymerase 1 does in a eukaryote? Makes the ribosomal structural RNAs. Paul 2 transcribes the protein encoding genes. And Paul 3, which is quite active, makes all the tRNAs, 5S RNAs, 7S RNA, some of these short nuclear RNAs. Paul 3. Those transcripts seem to have gotten turned into a mobile element. Now, Paul 3 transcripts do not code for proteins. They don't have any enzymatic activity. These things are completely dependent on something else to make them mobile. They got no transposase, no integrase. How does that work? It's thought that these short interspersed nuclear elements derived from Paul 3 transcripts are mobilized by lines. Lines mobilize signs. Always non-autonomous. They always require somebody to help them. Encode tRNAs, 7SL RNAs, 5S RNAs. These are transcripts by Paul 3. An example, be nice to know, is this ALU fragment. Well, I've talked about that before. The ALU family. ALU is a restriction endonuclease that when you cut the genome, you saw that it generated a band on a density gradient, a cesium chloride density gradient. You get a peak of a uniform density and form a shoulder or a satellite on the larger distribution of densities of the ALU fragments. And so it was indicative of a repeat that's there in hundreds of thousands of copies. Let's look at what, see, it's about 300 base pairs. That's not enough to code for a normal protein, and it doesn't encode a protein. It encodes nothing, and yet it's mobile and it jumps. It appears to have some homology and perhaps derived from the 7SL RNA. It is mobilized when it does move by a line. perhaps L1. So ALU is an example of a sign in the human genome. What's an example of a line in the human genome? L1 for line 1, L1. So L1 is a line. ALU is an example of a sign. May use lines to replicate. So this now begins to make a little more sense other than just the words rattling around in your head. Here you see all of these are retroelements because I don't have any retroviruses on here. The first category, the LT, are retrotransposons, TY in yeast, copia, and Drosophila. And there are some plant ones here, TN1 in tobacco, TUS17 in maize and so forth. But you have these examples, long-term repeats. The target repeats. This is the repeat it makes in the target when it jumps. They have reverse transcriptase and or integrase. Some contain introns, even. So the other class, these are the lines. Another name for a line would be long LTR retroposon. L1 is an example in humans. In mice, the lines are quite active. It's like, can't remember the percentage, it's like 8% or something crazy of mice. These things are jumping around in the mice genome still. L1 is the one in humans. They do not have the LTRs, target repeats, reverse transcriptose, and an endonuclease. We'll see how that comes into play instead of an integrase, and 1 or 2 ORFs. And then signs, and these are Pol3 transcripts. Doesn't code for anything, no introns, and these are very short, 300, 500 base pairs at most. These retrotransposons use RNA polymerase II as a promoter. Lines use Pol3 as a promoter. The signs, this is the tricky part. The RNA that they look like was originally a product of Pol3. But now, they're not really dependent on Pol3. They could be read by any promoter that's going to read them. So they're read by read-through, and Pol2 can do it. It's kind of unclear which promoter actually makes them, but they're derived from a Pol3 promoter. We'll keep it simple. We'll say Pol3 promoter. And here's another characteristic. The ones that are not retrotransposons, but the lines and signs, they have a poly A at the 3' end as part of the DNA. It's not added later, which means it's probably derived from a transcript. But this is odd because even the signs have a poly A, and these were originally Pol3 transcripts, which don't have poly A at the end. But through some contorted pathway, they now have an AT-rich region at their end. This poly A, and it might just be an A-rich region which has no evolutionary connection to poly A that's added on the end of transcripts, is used now to insert. It's at the 3' end, and when these two things here, the lines and signs, go jumping, they are looking for an AT-rich region, and the 3' end is doing the invading, and is looking for a little homology with an AT-rich region. So it may not be, it's certainly not related to ribosomes and translation like a poly A, a true poly A end would be. Here you see how they are related in terms of the homologies that they still carry. Here are the true retroviruses with the GAG-POL integrates an envelope, and then you see the pink here is GAG. You see GAG coming all the way down into these LTR, retrotransposons, TY and copia. And some of the Paul function, this would be the reverse transcriptase. They have LTRs on each end. But when you get down to the retroposons, not retrotransposons, but retroposons have no LTRs. There's just a little bit of enzymatic activity that's related to this Paul region. And it's reverse transcriptase still, because it still requires an RNA intermediate to move, and we'll see how that works in just a minute. But doesn't have a lot left. You have a reverse transcriptase, which also encodes in this ORF2 a endonuclease that cuts the target DNA. And right here in ORF1, you have a nucleic acid binding protein. Both of the products of these open reading frames, ORF1 and ORF2, have affinity, specific affinity, for their own RNA. So as soon as they are translated off the ribosome, they're looking for something to bind. In fact, they bind the very message that made them. They go back to mama. And that has implications in terms of replicating full length versus partial. And we'll talk about that in a minute, I believe. So you see, all of them share reverse transcriptase, because they all have an RNA intermediate. Retro, that's the retro part. Okay, so here you see the story about, see these two ORFs, this is the RNA that's made by reverse made by the host RNA polymerase off the DNA. And so this goes to the cytosol, translated on ribosomes, and the two proteins that are encoded bind to its own message. And here you see it binding to its own message. And this complex, now this RNA plus the two proteins travels to the nucleus and integrates. One of these is simply a RNA binding protein, just protecting that RNA. The other one, though, has the reverse transcriptase activity and the endonuclease to nick. It's going to nick the host probably in an AT-rich region. So we'll compare that with another mechanism. This means that this starts as a functional, this is derived from a functional line, one that can transpose itself, autonomous. This messenger RNA is fully capable of regenerating a full-fledged autonomous line. And notice it is protected and carried as a unit back into the nucleus. You don't understand the significance of that completely now, but that ensures that you bias replication towards functional units, because you're protecting that RNA that by definition is functional because it just made the protein that it needs to make. And when it goes back, it's in this complex and it can integrate. It has everything it needs to integrate this fully functional line. If this was let's say a DNA transposon, it's going to make the transposase, it's going to make a messenger RNA, it's going to go to the cytosol, it's going to make a transposase, and the transposase is not going to be attached to its mother RNA. It's going to go back to the nucleus, and it can bind to anywhere there's an end, an inverted repeat. So it could bind to a pseudo-transposon, a non-functional or a non-autonomous transposon, and move it instead of moving a fully functional one. So this is an evolutionary advantage that a line has because it encodes something that is cis-acting. It doesn't go in trans and float around and can choose which inverted repeat it wants to cut at like a transposase can. This is always linked together, so it's always going to bring that valuable package up here and integrate that thing in a functional sense. Lines, cis-acting. Propagation of functional elements is favored. And this is the mechanism, finally. I know you've been wondering, it doesn't have any LTRs. How does it do all this getting both strands made and switching the template back and all that stuff? It's still somewhat mysterious, but this is if you kind of peek through the spaces in the fence, we look in at the construction site, this is what we think we know. This is the target. And you've got this RNA, of course, which has the nucleic acid binding protein, and it has the reverse transcriptase and endonuclease all in one protein. And when it goes to the genome and it finds an AT-rich region, the endonuclease component cuts one strand at first. And that cut, the 3' end of the cut generated, serves to prime DNA synthesis. Here you see green, DNA synthesis. The purple is the RNA, and when it gets to the end of that original RNA, it now, and it's making, of course, DNA because it's a reverse transcriptase, it turns around and makes the other strand. Reverse transcriptase uses RNA to make a DNA copy. It can also use DNA to make a DNA copy. So it's the same enzyme that now uses the DNA that it just made to make the second strand of DNA. Of course, that happens, you displace the purple, which is the RNA, and you get, by some magic, you get to cut on the other strand, and you get joining and repair of gaps. And people say it real fast and kind of go like this. Everybody understand that? Okay, so lines have an endonuclease, and they are attacking with the 3' end of that RNA that's made from them, right? That 3' end is AT-rich. It looks like poly-A, and it's probably invading an AT-rich region of the genome and nicking using its, so that endonuclease, it's functionally integrase, isn't it? And then it has the reverse transcriptase activity. The new addition to the scheme is this nucleic acid binding protein, which has affinity for its own RNA. Retroelements comprise 40% of the genome. Things that do this kind of funny business, 40%. The good news is that most of them have not been active for millions of years. So you have retroviruses that we carry with us for millions of years. They haven't been active for 50 million. But we also can catch infectious retroviruses, right? HIV and lots of things. Transducing viruses. But these are not ones that we carry from generation to generation. Although we could, but they're not widespread in the population. Okay, so you see the percentages here. And sometimes I've seen on exams which is the most abundant retrovirus / retroelement in the human genome. Retroelements, we'll call them. Retroelements really, by the book, means excludes retroviruses, but the table here has everything. So you see actual retroviruses together with retrotransposons are only about 8%. The largest two categories are the lines and signs, and technically lines are a little more abundant than the signs. AL1, example of a line, Notice these two classes have the so-called poly A at the end, and 17 and 15% respectively. That's what most of this, the most repetitive element in the genome. Look how many copies of SINEs, the ALU, 1.5 million copies of that thing. DNA transposons, the regular old DNA transposons, about 3%. So if I ask you on an exam, I'll probably combine these together. I'll say, you'll see lines and signs lumped into a category. So I think that's the way it is on homework. And I don't try to break your brain because that's just 2% different. These are essentially about the same lines and signs, but they vastly outweigh the retroviruses lumped together with retrotransposons and the regular DNA-based transposon. And that's the end of what we cover this time. What are we doing on time? The rest of it I'd like to devote to review. Is it time to take a little 5-minute break? Okay, why don't we break and we'll come back and we'll cover some of the main points that you may need a little review on for the exam. That you have the camera on one side of you and it's elevated a little bit. One way you might do that is take a chair and clip it on to the back of it. I think this is what the guy in the picture did, but his chairs went up about this high. Another way you might do it is just take a little cardboard box and trim it down and clip the camera on the side of it. I'm trying to allow closed books to just be a stack of books. I mean, if you're going to open a book, you're going to have to take the camera off and look in. It's just ridiculous that they won't allow books there. Be inventive, but a little bit raised like this, and then I see kind of your face and the screen and the keyboard. I'm happy. Okay, so I'm looking over questions on homework in various places, and I see a homework question that I tricked myself. It's which of the following is true, and I'm going, I don't see any of them true. And I even put it in bold to alert myself that this is where the trick is. And the question was, which of the following was true in a series of statements, but the one that got me that was actually true that I didn't recognize as true was, it said recognized as false. It said, which of the following is false? And this is false. The beta clamp displaces DNA G in replication. You know, G is attracted to the helicase B. It makes a little primer, and then it's bumped off, and then the polymerase comes and elongates it. It's not the beta clamp that bumps off DNA G. It's the clamp loader that bumps off DNA G. Man, I'm so tricky. I may have to take this course again in the summer. Matter of fact, I expect I will. So I'm open to any questions, anything you want me to explain again. Of course, I'm not going to read the exam because the exam is put together each year from question banks, and I look and I notice that I don't have any questions in the exam right now. So that's what I'll be doing tomorrow, because it opens at 8 o'clock on Saturday morning. So I will be putting together a collection of question banks to give you 40 questions. And these are not as dependent on homework, but the questions are very similar. I looked at that website Quizlet and PCB 405 Exam 2, and I found that to be a good way to identify gaps in your knowledge as you kind of go through those. If you can do those with a closed book, make the little flashcard, but just right there online. So that's one way to do it. It kind of is more time efficient than going to the homeworks and taking them over and over. You've got them right there and you can quickly. So that can give you what's called surface learning. You'll get the A, you'll go to med school with that depth of understanding. But of course I throw in some critical thinking questions on occasion. You'll need to know more than that, but as a minimum, you need to know what the basic facts are. And looking at flashcards with a closed book is a good way to identify what you need to go back and sort of brush up on. So I think that's a very valuable exercise to look at those. For this last part on the retroelements and retrotransposons, the text, I think, fairly concisely describes the processes. So this would be one of those few occasions where I say you might want to go back and look at the text, and if you're tired of looking at me up here yapping, going over the videos, go back and read that text. I read that again this morning and found it to be informative. So you pretty much know the way I design a multiple choice quiz. I try to divide it into maybe 15 or 16 different question blocks that try to sample all of the major concepts and PowerPoints. So there won't be, even though you'll get random questions, you'll get random questions on the same 18 major themes. So everybody gets questions that they wish they didn't have, and questions that are really easy also. So I think it all sorts out. What I don't like is an instructor who has one question bank, and then you draw from that just by the Gaussian distribution. You can get heavily weighted into one area and almost nothing on another area. That's not good instructional practice. So I'm well aware of that, and I make sure that my questions don't overlap for the most part, and that you don't get overemphasis on any one part. So study it all, because you will get quizzed on all of it. And of course, study what you don't know. The stuff you hate, you've been putting off, it's time to look at it now. It's always that stuff you don't know. And before you study what you don't know, of course, you've got to identify what you don't know. You can't just have this happy feel-good feeling about going over slide till you get one you don't know. You should look at the notes and from kind of 30,000 feet, look down on them and pick out 10 worst slides in each PowerPoint. Try to distinguish between what's one you almost know and ones you do know, and those that you really need to study. And focus on those you really need to study. It's kind of common sense, but a lot of people take the approach, they'll just start slide number 1. Yep, got that. Slide number 2. Yep, I got that. I'm on a roll. Slide number 3. I got that. And I call that feel-good studying, because you're so reinforced with you got this is no problem. You don't realize how bad off you are. You don't realize what you don't know. So beat yourself up. Do all the hard, painful studying before you take the exam. So on the exam, you're not learning new stuff for the first time. That's not a good strategy. Also use a blank sheet of paper. I'm not much in favor of meticulous notes that you can publish and sell. I want those notes up here. So a lot of people get addicted to note taking. And it turns out, what I mean by that is they fool themselves in thinking that they know it because they have written it in such nice, legible script. And they've spelled all the words, and they got nice illustrating diagrams and all the little asides that I said, make sure you know this is on the exam. They have that, and they'll circle that. And none of it is up here. So it's better to have notes that you wouldn't show to anybody, because they're horrible misspelled words and the drawings look crazy. Nobody could understand them. that you can ball up and hide. You never did that. That's the kind of notes you want, because up here they look pretty good. Who needs more notes anyway? You've got the instructor's notes. You've got my notes. I drew all those little pictures on there and stuff. You don't need to draw anymore. Take a blank sheet of paper and see if you can reconstruct the stuff. But break it down into little narratives or little stories about a particular topic. What can I remember about DNA replication? And then, well, that's too big a topic. Why don't we just start with the enzymatic properties, the leading and lagging strand? What can I remember about it? Just start writing stuff down. That's a good way to force yourself to recall information. And that's what you're going to have to do in the exam, recall. Some of the questions that'll be just, oh, I recognize that as familiar, then that must be the right answer. But in some cases, you're going to have to dig a little deeper and be able to maybe manipulate the basic underlying facts. At least I hope that's you, or at least have some kind of challenge like that. Going to an exam review or forming an informal study group, especially with people you don't know. Because if you don't know them, you're not going to spend a lot of time talking about, catching up on all the events. And it's a little socially awkward, you don't know these people. In fact, you don't want to appear to be the only one who doesn't know anything. That peer pressure really puts your mind in a mode to learn if you know you're going to go to a group like that. Everybody just kind of underplaying their presence there. You've studied like crazy, but oh yeah, I just looked over this stuff, All that stuff helps you recall all those emotional tags that you'll be putting on those concepts. I like to organize groups with two little activities. One is come with maybe 3 questions that you're going to ask the other people you don't think they're going to know. And you are delighted in the fact that these questions you've got are quite clever. The person you ask is going to go, where'd you get that? And then go thumbing through their notes like this. I must have missed that. And that'll give you great joy and satisfaction as you watch the other person squirm. But you will have learned more than they'll ever learn from, because they'll forget the painful stuff. They're not going to remember that question you had. It'll be you that goes back still knowing the answer. But that's good, you know something. And the other thing, come prepared to explain something to somebody else. What I call a teachable topic. Take a process and know it thoroughly so you could explain it to your cat, your shoe, the wall, your TV, your GoPro. And kind of go around the group where one person explains their favorite thing. And of course, as you're sitting there listening, you're going, that's not right. You missed it. You totally missed that. It gives you that opportunity to criticize and stick the knives in, which is what you're really there for, to make the other person suffer. Because certainly you have suffered trying to learn all this stuff. So you can kind of game it, you can make it so that the brain is always interested in it and has emotional tags associated with concepts, which allows you to recall things a lot easier. So studying in a group is like the last phase. We can do some of that on Saturday. If you come, whoever sits at a table forms an ad hoc group. Everybody's there the same reason you're there. You're hoping you're going to learn something you missed. But the TA, her name is Nia, she's got a whole bunch of flashcards, and she likes to go from table to table, and she can kind of catch you off balance sometimes. I hope that's a productive activity for you on Saturday afternoon. And that'll be streaming also, so just like last time. Okay, any questions here?
Could you explain the difference between replicative and non-replicative transposition?
Yes. The question was, what's the difference between replicative and non-replicative transposition? Well, the first thing is, in replicative, you don't leave a hole when you jump, you leave the original copy. So that's the overall. If you do leave a hole, does the cell die? Does it like, I got a hole in my genome, I'm dead? What happens? Is that the end of everything? Anybody?
Does it just ligase itself back together?
Well, it uses non-homologous end joining. Q70, Q80, they don't look for sequence, they look for two ends and they go, oh, we can't have that. Let's put this back together if anybody notices. Not a problem here, nothing to see, move along. But when it does that, it can leave, it's not precise quite often. Like to get a, it's going to leave a repeat of itself. So you got that there. If it's in a non-essential region, it won't make any difference, but it could really mess things up.
To follow up on that, is just like one strand being removed? So like there's another strand left over, or is both strands removed?
Both strands go.
Oh.
It leaves a double strand, gaping, windswept, dark hole. And you look at, oh, a hole in the genome. Hope I don't fall in. Okay, so you want to know more about the mechanism, I presume. So you have replicative and non-replicative. In replicative, you have this strand crossover structure, this intermediate in the series of cut, snip, and join, that is also shared with one of the types of non-replicative transposition. And it depends on the order of cutting. In replicative, you cut on opposing strands, and then you take the donor, and it cuts itself at each end, and the 3' end at the end of the transposon goes and ligates itself to a 5' end at the target. And so at that, it is 3', it's ligating to 5', and the 3' from the target is now left to act as a primer to make a copy. And the same thing the other side of the transposon. The bottom strand now is the 3' invading strand, and it goes and ligates to the 5' end of that bottom strand, and then the top strand is 3', and that allows replication. So in that way, both strands of the donor transposon end up being primed from the target. And that is how you get two copies. And of course, that mechanism, when you get this replication, it ends up in a co-integrate circle that now must be resolved with resolvase, which is homologous recombination, but it acts at a specific site. So it's site-specific homologous recombination resolvase. And the site is RES, and that's an exam question on some people's exams. Yours. Does that, and with non-replicative, you get a cut on both strands of the transposon before it actually begins to join with the target. So it leaves a hole. What's a composite transposon? What makes a composite transposon move as a composite instead of as two independent IS elements? Why does it move as one great big joined composite structure? It contains mutations at critical regions. What's the answer?
The cut sites are outside, so they're able to carry.
Right. So each of my fists is an IS element, the so-called arms that flank the middle stuff. And each of these is an independent IS, but its inverted repeats have a defect on the inside copy. So you can't cut inside, you have to always cut on the outside inverted repeats of the arms. you need to know what happens when you get a transposon. Let's say it's replicative transposition. It makes a copy of itself, and usually these things don't jump very far, and it's got a copy of itself, 5 kilobases away. What does that open you up to? You got 2 copies of the same transposon in the same piece of DNA, whether it's the host chromosome or plasmid. Recombination, homologous recombination. And so you have 2 outcomes, depending on whether the 2 copies are in direct repeat of each other or inverted. And so the question always is, what happens to the intervening sequences? In one case, you excise what's in between the 2 as they recombine. In the other case, you invert what's in between the 2 copies. And the way I remember it is, if the 2 copies of the transposon are inverted with respect to each other, you end up inverting what's in between. Invert, invert. And then the other's just out. Direct, I say, you cut it out directly, but there's no real way to remember that. But it's easy to remember if you have 2 inverted copies, when they come together and you do the recombination, it'll end up not cutting it out, but just flipping the order of everything in between. Processivity. What is processivity?
The average length that an enzyme can go while it's before disassociating from its substrate?
Yeah, it's the tendency of an enzyme to remain attached to its template. It's another way to say it. So, does reverse transcriptase have a lot of processivity? Because it can jump off of its template and then jump someplace else. Often it'll jump to a second piece of RNA. It doesn't jump on itself very often, but it can. So it's low processivity. Can you think of a reason why that might be? Somebody told me the answer to this a couple of times.
Well, I know in like the high fidelity polymerases, it's a separate subunit that's used that partly grants it. Like the clamp, I think, is partly what gives it that.
It has no clamp.
Yeah.
That's one reason, but most polymerases don't have a clamp. Only one that does is Paul 3, but that's part of it. It has no clamp, and just it's evolutionarily beneficial. That's how it replicates. It deliberately has evolved a low processivity enzyme.
So it has a low processivity, so it can frame shift?
Well, frame shift refers to translation,
Yes.
but it can jump to another place using its new transcript sort of as a guide RNA. In the retrovirus model, it lands here and goes this way, but it goes over this region called R.
Oh, so it can,
And R is the same over here as it is over here, so when it floats away, it can go to any other piece of RNA, and if there's R on R, then it binds. Then it extends up. Yeah. So it's looking for homology of that transcript. It's sort of like Argonaut with guide RNA looking for a place to chomp. It's another question that I missed. It's the methylation effect on transposition. The question about TN10 and some of the eukaryotic DNA transposons, and it's which of the following is true kind of thing. I think this is from the homework at a minimum. It says, what's the effect? Well, let's just talk about it. Methylation inhibits transposon mobility. But it turns out it's a fully methylated state that inhibits. Sometimes, like TN10 and some of the retrotransposons become especially active just after DNA replication. And that's because their transposons are only hemimethylated at that state. So a lot of times methylation will interfere with the binding of control proteins, and methylation in a eukaryotic genome is sort of random. I mean, it's CPG, but these are distributed sort of randomly. If you happen to methylate a C, for example, that's in a protein binding site, let's say the transposase binding site, then you can inhibit its binding, but that usually is inhibitory in the fully methylated state. So some transposons become active just after replication because the DNA has not become fully methylated yet. Only one strand is methylated. Any other questions?
Could you go over again suppression frame shifting with personalizers?
Okay, so suppression and frame shifting, you have GAG, which is the structural proteins, the matrix, and then the capsid inside. You have two matrix layers. These are structural proteins. They're encoded in GAG. They all start with GAG, and it has a AUG, it starts, and it has a stop codon, it stops, it makes GAG, and that gets processed up. But on other occasions, the ribosome goes right through the stop codon into Pol. And you get a fusion protein of GAG fused to Pol. And there are two ways to make that happen. One is you simply suppress the stop codon. You keep the same reading frame. The stop codon is in the correct reading frame, except there's a very low abundant tRNA, a glycine tRNA, that has the anticodon for stop, for one of the stops. And it will go there and just put a glycine in, and the ribosome will keep on going and make a fusion protein. So that's one way. The second way is the ribosome will go along, and there'll be a particular sequence of RNA it's usually a stretch of phenylalanines, usually a stretch of A's, that destabilizes the ribosome, the translation process, so that the ribosome will get confused and either insert or go forward and get the codons out of whack, and it will shift the reading frame. And it will shift from one reading frame into the next. So it never really sees the stop because the reading frame got shifted, and it's just one continuous reading frame now from GAG to Pol. So suppression of a stop codon or frame shifting are both two strategies where you can take GAG and just fuse it right into Pol.
What's the significance of direct repeats and inverted repeats? We just really don't quite understand why they're...
Okay, so you get there's several ways we talk about repeats.
Yeah.
We talked about homologous recombination, whether it's inverted or direct. But there we're talking about the entire insertion element, not just the end sequence, the entire thing, because that's just homologous recombination. There's no cutting and all that. It's nothing the transposase is doing itself. So that's one context, inverted and direct. We're talking about the IS elements themselves, the intact, the full element. The other is the structure at the ends of every transposon. They have within them inverted repeats. And that's because in the mechanism of the transposase, it grabs one end and then looks for the other end and brings it forward. And now those two inverted repeats are lined up like that together. They now are in the same direction. And that has something to do with the geometry of the enzyme and how it cuts. But that's significant. And direct repeats, when it jumps into some place, when it cuts, the transposase not only cuts the two ends of itself at the direct repeats, like this, but when it goes to the target, it cuts the target in a staggered way. And then when that separates, we're going to separate them, then when the transposon comes in, you've got to repair a little bit here and a little bit there, and that generates a direct repeat of just the little nucleotides that are added, that are repaired. So that, it always makes a direct repeat.
If there are repeats, how can you tell what the staggers are?
Well, what you have to look at the sequence of a genome that does not have a transposon there. Look at its borders, and you'll see there's the first 9, 6, 4 nucleotides will be the same on the other side of it, that only one copy is there in the wild type, but it's not repeated. You just can't tell it from any other sequence of 4 nucleotides or 9 nucleotides. And that's the problem when it jumps, because it usually, when it goes back together, in order to restore the fully wild type condition, you have to remove one of those repeats. And there's not a simple mechanism for how to do that, how to remove one but not the other, to leave it in the original condition. That's why it's such a low probability event. Other questions?
So with DS elements in yeast, when it creates the breakage, we talk about like 2 chromosomes that can go into
That's in maize.
yeah.
AC and DS, breakage and fusion.
How does that actually happen when you have 2 chromosomes that are broken? How do they
Non-homologous end joining. If the ends of chromosome, these are just long linear pieces of double-stranded DNA, they're not circular, the chromosomes. There's a big tendency to fuse all the chromosomes together because they all have ends. And so the telomere, that's one of its big functions, is to hide that fact from the non-homologous recombination machinery. So it does everything it can to disguise itself. There's no end here, it says. No, you can, in fact, it takes itself and forms a big loop and splices itself back in, so you have the end kind of an eye loop thing. There's not an end. And back again, and go, where'd the end go? So it does all sorts of tricks with secondary structure. We'll get more into that, but to eliminate the ends. Because you really need to do that to protect yourself from transposons. And we'll learn about epigenetic mechanisms to keep 50% of the genome from expressing transposons, because one transposase that gets made could activate hundreds of defective elements, non-autonomous elements, and you get like whole herds like animals moving and transposing and jumping in and splitting and settling in. So there's a whole complicated machinery in animals and plants and eukaryotes to keep all of that suppressed, all locked up for millions of years. No expression for years.
So the reason why retrotransposons can't infect other cells is because they don't have spikes on their protein?
They certainly don't have the envelope portion.
Okay.
And that encodes for that little tower and the spike and the cap on it.
Yeah, that's okay.
Yep. Any more questions? If you have questions, you can get them answered several ways. The TAs love to answer questions. That's what they are there for, to answer questions. I love to answer questions. Just email me through not through regular email, because I get so many of those, you're liable not to get answered. But if you address me through the Canvas site, I will take care of it. So just in that inbox down there, because that goes to my email and I can tell, it's from Canvas. I know it's people who are going to evaluate me, so I must answer those questions. That's a good way. And then on Friday, tomorrow, starting at 1 o'clock until about 6, I'll be there to answer questions. Sometimes people come, they bring a chair, and we just throw questions. Somebody will have questions, and just kind of round robin. People will get questions answered that way. And I'm also available Saturday from 1 to 3, where I will take a look at the actual exam and make sure that there are not any things that are going to throw you for a loop. Because if you do well, it makes me look like a good instructor. So I want to effectively communicate to you what are the important concepts so that you can be prepared. Okay, that's all I have today. maybe I'll see you, maybe not. I have still some cameras that can be checked out. Just drop by my office, let me know you're going to come by, and I have maybe 6 or so cameras left.